Okay, so now we're on to life, sort of. You'll see that this actually goes beyond what you would think of as life, and then we'll go even farther beyond that this afternoon. But instead of me now chairing this festivity, you get my colleague, Immaculata DeVivo, who will be the discussant for this section with two speakers, Wally Fulweiler and Joel Dudley, who she'll tell you about in a second. But let me just tell you that I've had a fantastic time this year with Mac. The other co-director of the science program is Janet Rich Edwards, who's here somewhere, who's a Radcliffe Fellow this year. So we have a special co-director of the science program, my colleague and friend, Immaculata DeVivo. And when she's not doing her Radcliffe role, she's actually a professor of medicine at the medical school and a professor of epidemiology at the Icahn School. And she works on molecular biology, genetics, epidemiology, biostatistics, and she's interested in figuring out how those things can be predictive of people's cancer risk. But the reason you may have heard of her work and seen her on TV is she's one of those people who talks about the Mediterranean diet all the time. So over lunch, which is hopefully compliant with the Mediterranean diet, I don't think we asked her, you can ask her about that. But right now I'd like to turn it over to her, and Mac's going to tell you what's going to happen next. So.
Thank you. Thank you, Alyssa. Thank you. It is my pleasure to serve as the interim co-director for the life sciences here at Radcliffe. And life is broadly defined as things which are alive. And having looked at both speakers this morning and last night, and I said, well, gee, there's alive stuff in both of these talks, especially when Wally, who, as Alyssa showed, will actually cross both earth and life. But I don't want to take up the time going through the definition of what life is. I want to introduce these two fabulous speakers, Wally Fulweiler and Joel Dudley. And as you know, the bios are in your pamphlet, so I'm going to do a very brief introduction. Wally is, for the most part, an oceanographer from BU. She's a professor there. And her talk will focus on why the ocean needs you. And Joel will then follow with his talk. He's a professor at the Icahn School, Mount Sinai in New York. He will tell us that in his journey to this presentation or to this paper that he published, he was looking for drugs but found stupid viruses, quote unquote from the New York Times. So it gives me great pleasure to introduce Wally, and then Joel will follow, and we will have some questions at the very end.
Well, first, I want to thank the organizers of this amazing event for the invitation to speak here today, for Stuart for really getting us off to a fantastic start, and for that beautiful poem. And for you all, for the next few minutes, I'm going to torture you with my deep love of the ocean, and I hope that I inspire you along the way. So it turns out that I've been at Boston University and a PI of my own lab for 10 years. I know, you can hardly believe that. And I can hardly believe it myself. In preparing for this talk, I began to think hard about these last 10 years, where we're going and where we should be, and what are the major questions that we should be addressing. My lab addresses how coastal systems are impacted by humans. To do that, we use ecology and biogeochemistry, a terrible word, I know, but it links and tries to understand how biology changes and feeds on the chemistry and geology of the environment. Our goal is to understand how humans have changed these coastal systems. So we work across the land-ocean continuum, from forests and rivers to salt marshes, to your favorite bays and estuaries, to the continental shelf, and now into the deep sea. If you look at this slide here, this is one of the most, I think, beautiful slides of the Earth at night. And you can see where all the lights are, tends to be near coastal areas, because 40% of the human population lives within 100 kilometers of the coast. 16 out of the 23 megacities in the globe are coastal. So we need people to get energized and excited and interested in protecting these coastal environments because our humanity depends upon them. Over the next few minutes, I'm going to walk you through some different vignettes and ideas and little bits of, I hope, some sort of wisdom to inspire the next generation, and to talk to you about how I see science happening in the ocean world. But know that I think these lessons equally apply to whatever science you do. So first, behind me, you'll see a submersible named Alvin. If you don't know the submersible, go Google it, not right now, but later. It is a wonderful vessel that allows scientists to go down to the bottom of the sea, thousands of meters below the surface, to get a first-eye view of what is down there. And it is not just mud, although you'll see I'm a deep lover of mud, so it's awesome if it would be, but there's also wonderful findings that we will see. You may have heard before that we've only explored 5% of the ocean. That's insane. 15% of the ocean floor has been mapped by modern methods. So the majority of our ocean is an unknown world, and we need to get on with that. We need to solve that and figure out what's down there. In terms of discoveries, they're happening left and right, and they are major discoveries in the ocean. So for example, just a few months ago, brand new deep coral reefs were found off the coast of South Carolina, reefs we didn't know about. There are new fish. This is a hoodwinker sunfish. It's huge. It's a brand new species that was discovered just a few years ago. How did we miss that? Some of the genetic techniques that we're using allow us to pull apart species that would be harder. For example, Jeannie's dogfish looks a lot like other small dogfish, but it turns out genetically it's vastly different and a brand new species. These kinds of discoveries are the discoveries that get people's attention. They're sexy, they're charismatic megafauna. Well, maybe not this deep sea fish. That looks like chewed bubble gum, but the idea is there, right? It's a deep sea fish, it's exciting. But these are not the only parts of science that matter. Additionally, as we move forward, the technologies that we have are growing, and these technologies are allowing us to see and discover things that we have not found before, and the pace of this discovery is only increasing. This is a brand new sequencer called a Minion. You know, if you want to send me a couple, it'd be great. And this allows you to sequence genomes in situ. You can take your sample on board your research vessel and sequence it there. You could do that in the salt marsh from your car if you wanted to, if you had a big enough car. Anyway, so it works. These are Argo floats. These are floats that are, all of them, almost 4,000 of them, a global effort to understand the surface currents of the ocean. Each of these floats is beaming back information about how fast currents are flowing, their salinity, the temperature of the ocean. Many of you likely have a smartwatch, right? It's telling you your heartbeat and your sleeping rhythms. Well, let's do this for the ocean. We're there, we can do this. And the rhythms in the ocean are profound. They don't have a heartbeat, right? But we have tidal, we have ebb and flood of the tide. We have huge animal vertical migrations, moving from the depths during the day upwards at night to feed. We don't know a lot about this, and we need to understand the amount of organic matter and organisms that are doing this and the impact that that has on our ocean. Here I'm showing you what I think is a pretty neat diagram that my PhD student, Emily Chow, made. And you can see here that it's showing you how we can make a smart ocean. We can outfit human divers. We can have different systems on pump profilers. We can have the submersibles like the Alvin I showed you, different cabled observatory. These things are happening, they're coming online, and we need good minds to be working on these data. So whether you're interested in the science or the engineering or the computational side of it, the ocean needs you. One of my favorite stories that I'll just quickly touch on here is this piece of instrumentation here is a flow cytometer. It's mainly, traditionally it was used in the medical field until Penny Chisholm and her group at MIT used it to measure phytoplankton cells in the ocean. They went to areas of the ocean that they thought didn't have a lot of productivity, and this was about 30 years ago or so. They applied flow cytometry in a brand new way, and they started to measure all these different phytoplankton. And then there was this like pesky noise that kept popping up, and they ignored it, because that noise signal that was coming through was way too tiny to be anything. Until they slowly realized that that noise signal seemed to be changing with things like salinity and temperature, and perhaps it wasn't noise at all. And in fact, it wasn't. It was this beautiful little tiny green photosynthetic cell, this tiniest photosynthetic cell we know of, named Prochlorococcus for little green berry. And Penny Chisholm and her group transformed our understanding of how the ocean works by taking new technology and applying it. And in fact, this is the harbinger of, came from, I should say, the cyanobacteria that gave us the oxygen that we enjoy breathing. And so this tiny, tiny cell, using a brand new method, they found it and changed how we understand the ocean. Lest you believe that you must have giant discoveries to make a contribution to science, I want to make a very important point, and that is that all discoveries count. Small discoveries are important. It is the small discoveries that allow us to add drop by drop by drop until we fill this pool of knowledge and ignorance, as Stuart would say, that allow us to increase our understanding of the ocean environment. It is not every day that you discover a new species or find out about a brand new tiny photosynthetic cell we didn't know about, or get to dive to the bottom of the ocean and find deep sea coral reefs. Those are wonderful, magical moments that get lots of attention on TV and social media, but they are not the everyday sort of work and discipline of science. So it's important to know that all discoveries count. I want to tell you a brief story about when I was a PhD student and how I learned and continue to remind myself how important it is to be skeptical and challenging of the work that you do and others do, but also to have faith in the work that you do. That if you are a good scientist, you need to have faith in the work. So one of the things that I am deeply in love with when it comes to the ocean are sediments. There really aren't that many good pictures of them, so I've made you this beautiful slide to show you. There's our water column and sediment. It's oxygenated up here, and then it's not, there's no more oxygen when you get to certain depths in the sediment. One of the things that the sediments do is they process organic matter and carbon. They recycle nutrients for us. Sometimes they produce greenhouse gases, sometimes they consume them. And they have one in particular thing that I'm interested in is they have an ability to remove nitrogen. Now, nitrogen is fundamental to life. Without it, there would be no you, no me, no cod, no blue whales. But like anything, too much nitrogen leads to problems, right? Excess phytoplankton blooms, harmful algal blooms, low oxygen conditions. And where there are humans, and maybe you remember from my earlier slide, there's lots of humans on the coast, then there's more nitrogen. And that nitrogen then can degrade ecosystems. But there's a natural filtering process called denitrification that microbes do. And in coastal ecosystems and in marine systems, most of those microbes live in the sediments. And what they do is they take a biologically usable form of nitrogen, say nitrate up here, and convert it to N2 gas, which then can go up into the water column and into the atmosphere. Our atmosphere is about 70% N2 gas. It's an inert form, okay? So the key to remember is that I was really interested in this process because I wanted to see how the natural filtering process in sediments was removing nitrogen. And what I expected to see was that I would measure N2 gas being pumped out of the sediments, right? Think of the sediments as exhaling this N2 gas, and I was going to measure this. And I did it in a coastal system in Rhode Island known as Narragansett Bay. And I did it there because the system had been changing due to changing climate, and there was some idea that maybe the nitrogen removal capacity of this system was changing, and I wanted to quantify that. I had this wonderful historic data set to build on because the first measurements in denitrification in a marine system were done here. So I could say, okay, well, this is what the denitrification, right, the N filtering process looked like in 1979. Lots of N removal. Oh, 1986, we still had lots of N removal. And then I came along in 2005, and we still had N removal. Great, it was a little less, but it was happening, and this is good. At this point, I was doing all of my work measuring the actual fluxes in a well-known lab in Woods Hole, the Marine Biological Lab, and Ann Giblin's lab with Jane Tucker training me. And so I felt confident in this because I was, working with the best. And then I helped my advisor get funding for this instrument, so Cadillac of mass specs, in case you want to know. If you're interested in measuring dissolved gases in seawater, this is the model you want. Happy to talk to you about it. So we wrote a grant, and he wrote the grant, I wrote part of it, this part, and we got this mass spec. And boy, was I excited. We set up the mass spec. I go get my sediment cores. I'm getting out there to measure the next year. And I'm like, oh, I'm going to measure this N removal, all this N gas coming out. And yes, it's going to happen. And bam, this is what I found. The exact opposite, not what I expected. I thought maybe it would be the same. No, I brought it to my graduate advisor. He said, did you wire it backwards? Did you run the samples backwards? And we methodically went through all the things, and I won't tell you those details, but just to know that we didn't wire it backwards. I didn't run the samples backwards. And now what we have seen is that this system actually flips back and forth between removing and turning it into a gas and actually making nitrogen. The bacteria is sucking nitrogen in from the water and turning it into a biologically usable form, which has impacts, higher trophic level impacts. So this I learned. One other thing I want to note is that when I started to go around to conferences and give this talk, many people came up to me and said, I had these results and I sat on them because it didn't fit, and I'm going to use air quotes and I apologize, the working paradigm. I want to ban working paradigm from science because I think it squelches ideas and progress. And so now it turns out that many ecosystems have seen this because we begin to slowly pull this away and realize that just because it goes against the working paradigm doesn't mean it's wrong. I want to mention how important it is. This is a library for the students in here who don't go. It is now a criterion to graduate from my lab. You must go to the library and send me a picture of you reading a book in it. I deeply worry that we're going to be losing information that's not digitized because people don't go to the library, but you should go. It is wonderful. And you should take time to read and to write, which really means rewrite over and over again, because there is a vast knowledge, and unless you prepare your brain for that discovery that might happen, you may miss it. The prepared brain, right, is the one that will be open for those opportunities. And there's only one way to do that, and that is to sit in the chair and do the work, the drudgery a little bit, the less sexy part of science. Mm-hmm. It's also deeply fulfilling. I want to mention that persistence is key. It is vitally important to know that when you fail, you must keep going, and that all of those small discoveries, right, they build to bigger things because you are persistent. So be like the waves on the shore. They constantly come. Sometimes you will work in a fury of a hurricane, and that is a wonderful feeling. And sometimes you will work slowly, ponderously, and that is fine. The other big waves will come. So just be persistent. We speak often of eureka moments in science, and I must tell you, I know of only one in ocean science, and I have had the fortune of actually making sure in person that it was real. I got to talk to Dr. Colleen Cavanaugh about this. The eureka moment story goes like this. These are riftia. They are giant tube worms, sometimes 6 feet tall, found at the bottom of the ocean at hydrothermal vents. They have no mouth, no gut, no anus. When they were discovered, nobody understood how they worked. There was lots of work kind of pulling them apart and looking inside the side of them and trying to figure this out. Dr. Cavanaugh was a first-year graduate student at Harvard. She sat through numerous seminars on this, and sitting in one of those seminars that I understand was quite laborious and long and somewhat boring, she realized during that seminar that the way these animals work, since they have no insides to do so, must be a symbiotic relationship with bacteria. The story goes that she actually shouted this out, I know what this is, it's symbionts, right? It's sulfide oxidation, these bacteria can do this. This is one of the few eureka moments, but if you think about it and you talk with her about it, and I hope that you do, she'll tell the story better than I do, you'll learn that she had done a lot of previous work. She was broadly trained in ecology. She had been working on microscopic techniques. She had been thinking about bacteria and symbiosis. So the stage was set for her to make this discovery, and that goes back a little bit to this idea that sitting in the chair and reading or doing the work in the lab, they all build on each other. Persistence is key. I want to speak, too, about what motivates you. You need to find it and pursue it with love and passion and energy. These motivate me, right? Yeah, it takes all kinds. So these are small plastic bottles full of ocean water. This particular set is from Narragansett Bay. It's where I grew up. I grew up on this bay, and I thought I knew it. I thought I knew all of it. I can tell you when the southerly comes in at lunchtime. I can tell you where to swim. I can tell you the ebb and the flow of the tide. I can tell you how to skirt along Dutch Island and not hit the rocks. I can tell you the creepy spot if you want to go swimming that I'm pretty sure there's a big scary animal down there in this one area, right? I went to graduate school thinking I knew this bay. And then I started sampling. And we collected these bottles to study the nutrients, so things like nitrogen, phosphorus, silica, the fertilizer for phytoplankton, for the grass of the sea, the elements that don't get much love, and yet without them we would cease to function. We collected these samples, we brought them back to the lab, and all of a sudden these data came out. Concentrations of nitrogen, concentrations of silica. And with these data, I could tell a story and see a snapshot of a bay that no one had seen before. I could tell you about life and death of animals. I could tell you how a warming climate was changing the bay. It is these plastic bottles, this little tiny plastic bottle, that transformed and changed my life. And at 3 in the morning, when I wake up to think about what's happening in the ocean, I often think about the data, or the data that are coming out of these bottles. It's super nerdy, I know, but embrace that nerd, because we need you. Finally, I want to say 2 more things. I want to say that you can't do it alone, and you shouldn't do it alone. And if you get to a position where you have found the thing that inspires you and you get to work with that for the rest of your life, then build a team around you, the team that you want, and bring others with you. Here I am showing you some of my graduate students. I want you to note that and postdocs, I want you to note that they are smiling. They are covered in mud. They are working in gross chambers. This is like hours of filtering. For hours, and that's just the filtering. That's not the lab analysis or the data analysis. They are covered in mud. They are working on almost intractable problems, not because they will solve them. Here's Emily working on underwater mass spec so we can measure the breathing of the ocean seafloor. These are happy students because they have found what motivates them. So find that, get others to come along with you. Finally, I want to have a shout out to people that support us. And importantly, I want to tell you one brief thing. Growing up, I have two older brothers, and I have a super supportive family. I'm very fortunate for that. And one thing my mother would always say to us is, you could study this tidal pool your whole life and never know everything about it. And it kind of became a family joke, where we would say, like, hey, Mom, see that puddle? Could I study that my whole life, right? Well, it turns out that she's absolutely right. That this tidal pool represents this amazing ecosystem. There are animals here dealing with giant waves, managing to live through waves hitting them, right? Desiccation from the noonday sun. There are neat bacteria in here, different crabs, seaweeds, macroalgae, so many things. My point is to tell you, you need to find your tidal pool. Go after it, because we need you. The ocean needs you. Thank you.
Fabulous. Great. Just 1 second, Joe. Thank you. Thank you, Wally. That was great. And I think this was the second time that a mom was invoked to being right. Let's hear it for the moms out there. Yeah. That was great. Lots of questions, but we're going to have Joel tell us about those pesky viruses.
Okay.
And then we're going to open it up to questions.
All right, thanks for having me here. It's great to be here from New York. I was getting a little nervous before my talk, but I heard during Wally's talk someone honking and screaming at somebody, and it made me feel at home. So I feel much calmer now, so it's good. So I'm going to talk to you about unmasking thieves of our health hiding in the shadows of biological understanding. 2 simple questions really motivate a lot of my research. What is health? What is disease? I'm going to argue we have very, very unsatisfactory answers to both of these questions. But first, before I get and talk to you about the research that I do, I need to sort of orient you in how I think about the world and how I approach biological research. And that's first by appreciating there are rarely smoking guns in human health. What do I mean by that? There's rarely the magic gene, the magic protein, the magic drug target that's going to be the solution to your problem if you're studying disease. What we're dealing with for the most part is smoking gunships. And I'll give you a great example of that. I have Crohn's disease, inflammatory bowel disease. Genetic association studies have found over 200 genomic loci associated with Crohn's disease. You could loosely think of that as 200 genes being associated with inflammatory bowel disease. By the way, those 200 genes don't yet explain 50% of the genetics of that disease at this point. So who knows, when things all said and done, we'll have 400, 500, 600 genes for that disease. And where's the drug target in all of that? So, and it really shouldn't be surprising that, that things are so complex when it comes to disease biology, because humans are complex, layers of complex networks all the way down. Let's just think of a single cell for a second, right? A single cell in the body has 3 billion base pairs of DNA that are wrapped in this sort of a spool and thread around these chromatin states. 3 billion base pairs of DNA express some RNAs, and you might have learned in high school biology DNA to RNA to protein, but that's like the Rutherford model of the atom, actually. It's actually DNA to probably tens of hundreds of different types of RNAs, eRNAs, picoRNAs, link RNAs, the list goes on and on. We're still discovering different types of RNAs. Those RNAs go back and bind to the DNA, regulate DNA. Then some of those RNAs create proteins. Proteins form complexes. They go back and bind to the DNA, regulate the DNA. So just inside one cell, we have these different layers of networks interacting both with themselves and across these different layers. For example, protein to DNA. And we haven't even left a single cell yet. We have a whole constellation of interactions in a cell. Cells form tissues. There's cell-to-cell communication. Tissues form organs. There's organ-to-organ communication. And if everything we had in that box was all we had to worry about, life would be simple. But the environment interacts with this system. So humans are complex adaptive systems, which is sort of a well-understood type of system, and at least in how we think about it in, say, physics or mathematics. Complex adaptive systems have key characteristics as they evolve over time. There's interactions like I just talked about. The whole's greater than the sum of the parts. And here's the key thing. You can't understand the system by looking at its parts. But what do we continually try to do in biological research to even today is try to understand the system by looking at its parts. But we know we shouldn't be able, we can't really do this in a complex system. Who said this, by the way? This is an investment banker in Harvard Business Review. So for as much grief as we give the bankers, they have a pretty good understanding of complex adaptive systems. So being a patient myself has helped me think of new questions about disease. This is a recent trip to Greenwich Hospital. Now, I did not put this picture up here to get sympathy. Now, don't feel bad for me because I'm so loaded with morphine in this picture. I feel 100 times better there than I do on the stage right now. So this is not to get sympathy, trust me. You just can't tell on my face because I'm somewhere else. So one thing that happens when you get inflammatory bowel disease is there's cutaneous, which is an immune disease of the colon. You get cutaneous manifestations of disease, erythematosidum, and a lot of skin rashes. So this happens to me all the time. Whenever my disease becomes active, I get a rash on my chest. So I go to the GI doctor and I say, isn't this interesting, that my skin reacts when my bowel disease is about to, act up. And he said, oh, we know about this, of course. And I said, well, what do you do about that? He goes, well, we send you to the dermatologist. And I said, well, there's a connection there. And so I go to the dermatologist and I say, well, isn't this interesting? My skin is connected to my gut. It's kind of gross, but your gut's kind of your inside skin if you think about it. Like, what do we think about the biological connection here and what it means to understand these diseases? And the dermatologist says, I specifically became a dermatologist to never, ever think about the colon. Please take these steroids and get out of my office, right? So being a patient made me realize I was stuck between these two systems. So one of the things we did early on, I did as a result of this, is I took all of the emerging genomic data that was being generated. This is actually when I was a graduate student at Stanford. And we asked the question, does the molecular taxonomy of disease look anything like the symptoms and anatomy-based taxonomy of a disease that we still organize our health systems and our research institutes around today? You find when you sort of look at the genomic patterns in terms of diseases genetics that are being disrupted between diseases at a genome-wide scale, you find, for example, that at a molecular level, Alzheimer's disease looks very similar to actinic keratosis, which is a skin disease, which seems kind of bizarre until you remember that the brain and the skin are together at one point in a structure called the neuroectoderm in development. So using data to redefine the landscape of disease, we can actually take this concept up to the patient level. One of the first things I did at Mount Sinai was getting access to a huge amount of electronic health record data, as well as some records where we have DNA, is I wanted to put into place this idea of a learning healthcare system, which is kind of a buzzword today. So what frustrates me still is when I go to a health system, they'll say, okay, Joel, you're a 40 to 45-year-old Caucasian male, family history of this, complaining about this, let's treat you based on guidelines we know about this situation. And as a data person, I see, well, you see millions and millions of people literally every year. Which patients look like me and what happened to them, right? So like when you go to Amazon, all the products that are served up to you are based on data of people like you, for example. So what you're looking at here is not a network of diseases now. You're looking at a network of patients. And what I did is I took all of the quantitative data we had on every patient in electronic health records, heights, weights, metabolic panels, CBCs, et cetera, and I represented patients by all the quantitative information we had on them. Turns out to be 200 or so. But I ignored whatever the diagnosis codes are given by the physicians. I considered those to be subjective. And then I said, which patient looks like that patient, looks like that patient, looks like that patient. I mapped out this sort of map of patients. I know from last night there was an algebraic topologist in the audience. I used algebraic topology to map this out. So I don't get to meet one all the time. So, but the idea here is this. So if I'm a new patient at Mount Sinai, is my little Waze or Google Maps stop blinking here? Is it blinking here? And does it really matter, right? If I'm here or there. So what we showed in the paper, which I don't have time to go into too many details, is you definitely want to be over here. You don't want to be over here. I call this cardiometabolic island. It's about the nicest name I could come up with for it. But the color here is important. And then after the patients were sort of organized by their quantitative data, we said, where are the patients with type 2 diabetes? And because we have lots of them here in the United States, we're statistically powered to look at them. And these red zones are sort of hotspots of type 2 diabetic patients. They're not all in one spot. Type 2 diabetes is obviously this disease we sort of think is a single disease. It turns out if you just focus on the type 2 diabetes patients alone, you get these three very distinct clusters of type 2 diabetes patients. And the provocative idea here is that we have type 2, type 3, and type 4 diabetes sitting under our nose the whole time, but we didn't know it because we never looked at our patients in this data-driven way, right? So you can cluster data all day long and get cool figures, but who cares, if it doesn't mean anything clinically. So we had many of years of electronic health record data on these patients, and briefly I can tell you we showed that patients in this group, type 2 diabetes patients, had increased risk of heart attack and stroke. So if you're a diabetic, you have increased risk of heart attack and stroke, but if you were in this group, you had risk above and beyond other type 2 diabetics. If you're in this group, you had increased risk of cancer. Now all diabetics have increased risk of cancer. If you're in this group, you had risk above and beyond the other type 2 diabetics. Now we also had genetics on these patients. The genetics was not used to inform the clustering, and why this paper was published in a pretty decent journal was we did genetic associations on these subtypes. And remember, since the genetics didn't inform the clustering, it was independent. And we found genetic markers that matched the differences in disease risk that we found. So that's harder to explain away the fact that the genetics, which was independent. For example, in this group, there are genetic markers that were found in vascular endothelial signaling pathways. So again, suggesting that there are these subtypes and even markers which we could start to bin patients into. So the thing I love about data-driven approaches is it saves us from what's called the streetlight effect. Right, this kind of cartoon is a cop is asking, why are you looking for your keys under the street light? And they're saying that's where the light is brightest here, right? So we didn't coordinate on this, but I have Donald Rumsfeld in my slide too. So, because I think he said it a little bit better. So if there are any journalists here, right? So here's the headline I want to see tomorrow, is scientists gather at Radcliffe Institute to celebrate scientific legacy of Donald Rumsfeld, right? So. Because it's coming up, right? So please, let's see that in the paper tomorrow. Yeah, so because clearly that's what's happening. So I think a more recent example of where we applied this data-driven approach and came to this very unexpected and shocking result was in Alzheimer's, and which Immaculata alluded to. I got one of my favorite quotes ever in the New York Times. I went looking for drugs, and all I found were these stupid viruses. So I'm going to go through how we got to this unexpected discovery. So if you're not aware, Alzheimer's is actually a very complex disease, and we don't know everything about the pathophysiology, etiology. But for the most part, we're focused on, especially for drug discovery, the formation of these amyloid plaques, and also these tau tangles that can form inside the neurons. And for the most part, drugs have focused on clearing beta amyloid. You can actually do amyloid scans, and you can sort of see the aggregation of this amyloid protein in Alzheimer's disease patients. So again, the drugs have, for the most part, focused on can we get rid of this amyloid. Since it's a characteristic pathological finding of Alzheimer's disease, turns out that hasn't worked very well. There's effectively a 99.5% failure rate or something like that of Alzheimer's drugs. I mean, we failed over and over again. This is the number of compounds that have failed since 2012, and the number of trials that had failed. Now, it would be great to say, well, since 2012, things have gotten better. It stayed exactly the same. So, and it's really gotten to this point of desperation now, where you have these articles, are we wrong about the theory of Alzheimer's? And is the leading theory wrong? In fact, our study, when it was published, there's a software tool that our media folks used that suggests 800 million people read about our story in one way or another through their media, which means we're sort of at the end of our rope and we're hungry for something new in Alzheimer's because we're kind of out of ideas. So we leveraged a resource that the National Institutes of Health had generated. It's actually a very forward-looking resource. It's called the Accelerating Medicines Partnership for Alzheimer's Disease. And really what it is the biggest multi-omic study on Alzheimer's patients ever really conceived and executed on. So you're taking many, many layers of complex data collected from patients with, obviously, post-mortem with controls in Alzheimer's disease, genetics, genomics, metabolomics, every ohm you can think of, they're measuring in this study, and even layering in clinical data, et cetera. So and we were actually invited in by, again, a very forward-looking program officer who realized that most people are going to be looking at these slices independently because they're experts in this area, and they wanted to bring in people that are going to look across all of these, right? So there's just not too many people that do that. So how do we look at this type of data and sort of this holistic modeling approach? Is we use something called graphical modeling. So if I can so imagine, that, this is a genetic marker, this is a protein, this is a metabolite. The other thing might be a pixel and a brain imaging scan, and we sort of look at all these data together, and we see, well, every time this genetic marker is varying in the population, this metabolite's going up, so we'll draw a graphical connection sort of between these things, and sort of map all these layers of data in this sort of multi-scale network model. And then we actually have tricks to turn these from pure correlation networks into sort of directed networks using like Mendelian randomization and things like that, so we can even understand how information flows sort of from parts of these multi-scale networks and to the other part of the network. So the question we were really trying to ask was, well, let's not just focus on beta amyloid, which is really the predominant theory in terms of drug targeting. Let's look at these very complex networks that we can construct from all this multi-scale data and look at the healthy brain network, again, genetics, genomics, proteomics, et cetera, and the Alzheimer's network, and let's just see how they're rewired. Let's figure out how they're rewired from the healthy to the disease, and that might reveal some novel drug targets. We could target other aspects of the physiology that's transforming the healthy brains into the Alzheimer's brains. So this is actually a highly simplistic version of the software and the computational steps that go into doing that. I'm not going to go through this, but needless to say, it's actually something that runs on a supercomputer. It takes a couple days to a week to get the answer. So we were crunching on the data, and really what came out was, as we started analyzing the genes and the networks and things that were getting most rewired, you can think of it this way, between healthy and Alzheimer's, a screaming signal for viral biology. And we said, oh, crap, right? Because that is not what we were looking for. I was looking for drug targets. I was looking for things like GPCRs, ion channels, and things that are known to be druggable. But what we found is a screaming signal for viral biology. So we said, well, now we have to go back and look at this. And it turns out that the standard software for analyzing genomic data, which actually is created here, actually throws out viral sequences by default because it's often contamination, right? So we had to go back and sort of rewrite our own software. So even another person looking at the same data set wouldn't have found the viruses. So what we're able to do in the end is pull out these networks where we can actually see viral genes. So one of the top viruses we found sort of directly interacting in the core networks that were driving from healthy sort of to Alzheimer's pathology were HHV6, HHV7. These are actually a type of sort of herpes virus called roseola virus. And HSV1 also had been implicated in the past by some folks here as well at Harvard. But what's different this time, instead of finding correlations, we could map specific biological networks in which these viral genes were directly interacting with the host human genes, such as PSEN1, which is a very well-known Alzheimer's gene. So we get very specific testable networks or biological sort of pathways that can be tested about how viruses are contributing to Alzheimer's. We even discovered a novel gene such as this miR-155, which actually is a viral response gene, which we showed in this animal model increases beta amyloid plaque deposition. So what did we discover? So we discovered that these common viral species that are frequently found in the brain can in fact activate and drive, we believe, Alzheimer's pathology, and particularly HHV6, HHV7. So, and what's really notable about this is we actually replicated this in different cohorts. So this AMP-AD partnership is they bring in brain banks from several different institutions. So we're able to replicate these viral findings across independent brain banks. If it was just contamination, it probably would have only been found in one brain bank. And we actually identified specific testable biological pathways, which we can sort of follow on this. But I actually want to point out a couple things that aren't on here that I'm really proud about in terms of what this sort of unexpected discovery led to, which is actually related to the prior talk. So one thing that happened was I started giving talks at National Institutes of Health and other places about this viral finding, was a bunch of intellectually closeted researchers coming up to me saying, oh my God, I've been sitting on data for years that totally supports this, but I've been too afraid to publish it. Why? Because their whole career is in the Alzheimer's field, right? So I have ADHD, I do a bunch of other things. But so I could care if they hated me or not. So, so this happened every time I gave a talk, another researcher came up and said, oh my God, thank you for paving the way for me to finally be able to publish this data I've been sitting on for years. So again, it's a huge problem that I didn't realize happened in other areas. Another great thing that came out of this, of course, was the fact that there was a quote from the NIH, I think, officer in either the NPR article or the New York Times article, where they sort of said, and I'm editorializing, well, darn it, I guess we're going to have to start funding research in this area because there's such an overwhelming amount of data. In fact, the New York Times article, New York Times person said they're trying to find someone to speak negatively against our article, because that's what they always try to do and they said they found someone that truly hated everything about our paper and spent the entire weekend trying to find something terrible to say about it. But the best compliment we got was they said they dropped the mic on the data analysis and I have nothing to say on the paper. So, and of course we put all this, and notable is we put all of the data and all of our software, all of our code is in the public domain for anyone to reproduce. So when we give talks about these scientific findings, a lot of people think, well, how does it relate to me, for example, if someone's sitting in the audience? So, well, if you're interested in Alzheimer's, there's all kinds of cool stuff that we're following up on, such as using antivirals to treat Alzheimer's disease and a bunch of new potential druggable pathways we've identified. But I want to point out something else we're doing to really take the spirit of what we did in terms of analyzing the Alzheimer's data and taking this data-driven approach to illuminate what we don't know about health and disease, down to the individual patient level in a way that you could personally experience. So we have this project at Mount Sinai called Lab 100. So the practice of medicine, I mean, talk about not embracing the undiscovered. Practice of medicine, I would argue, is among the worst at this, either by trying to mitigate the undiscovered or pretend it just doesn't exist, particularly by evidence-based medicine, right? So they like to focus on evidence-based medicine. Of course, a lot of this evidence is based on small studies of a couple hundred, if not 1,000 people. And, and we really try to mitigate discovering, incidental findings and things that are totally unknown and trying to find ways to deal with it. So what we're building is this new type of clinic at Mount Sinai that's actually going to open the doors soon to really embrace the undiscovered as part of routine medical care. And we're asking the question, what if you actually had a health record? We always talk about electronic health records, but we don't have health records. We have records of your diseases and your treatments. Health is currently defined as absence of everything we agree is a disease. If you think about that for a long time, that's pretty embarrassing, right? I mean, that's our definition of health, right? So if we had an electronic health record, what will we measure, right? And we measure all these different aspects of your health. I'll give you a sneak peek at what this actually looks like. This is when you actually start in the clinic and you have your initial consult with your doctor. And we do everything you're not supposed to do in medical care, is we measure thousands and millions of things about you and every aspect of your health when you're not complaining about a disease, right? I mean, that's enough to give a physician who went through the standard training a heart attack, right? So, and there's good reasons for that, but data can deal with incidental findings. And this is what it looks like, and I'm not going to show you the in-between because I ran out of time, but this is what it looks like at the end of your visit. We do a whole 360, highly data-driven, millions of data point view of your overall health. We have a conversation with you about it, and we give you access to this, and you own this data. So we're actually building clinics that embrace the unexpected about each and one of you, because there's so much we don't know, right, about health, about wellness, about vitality and resilience. It's waiting to be discovered in each and every one of you. So thank you.
Thank you.
Yeah.
Yeah, I'm going to talk with that for 10, absolutely. Well, that was great. Thank you, Joel, and of course, thank you, Wally. So I'm going to just, if you will, start the questioning, because I actually have a lot of questions related to both talks, and then we'll open it up to the audience. So I'm going to start with Wally. So I'm going to go with the glasses half full. So we don't know much about oceans, right? Mm-hmm. And can we say that's partly driven by the fact that we didn't have the technology, the instrumentation to actually go looking for this? And I was provoked to ask that question, given that beautiful green little thing that you showed by your MIT colleagues, mm-hmm. so that we wouldn't have been able to find that. So it's sort of like one of these questions where, and it also applies to Joel somewhat, is that sure, we don't know what we're not looking for, but a lot of it is driven by what we can actually look for. And there is a Rumsfeld quote that I want to use, given that Rumsfeld is playing such a prominent role today, which is, you cannot go to war with what you want, you go with what you have. And I know I mangled it completely, but sort of the spirit is there, and it sort of relates to these questions that both for you and for Joel, but Wally, you can go first.
Sure. So I would say to Rumsfeld, you probably shouldn't go to war then, but sorry.
Just a quote.
Just a quote.
No politics.
Sorry, no politics.
Just couldn't resist that quote.
So I think that for sure our technology is increasing at this really rapid rate. I mean, Alvin's been around for a while now, but it just got a major retrofit to go to deeper depths. Please don't ask me which depth, because I currently forget. But I do think our technology is really increasing at this fantastic rate, and I think this is a wonderful time to get into science in general and into ocean science. And I think it's really important too, because for example, the finding of those coral reefs off the Amazon or off the Carolinas, we have ideas about trying to do things like deep sea mining or drilling for oil, and we may be making decisions, or fishing, we might be making decisions without fully understanding the environment that we're going to impact. And since we so rely on these environments for the well-being of humanity, I think it's not only important that we're using this technology to explore, but it's actually, it's not life and death, but it's vital to humanity to do so.
I'd like to capitalize on that, because in the short term, it doesn't look like life and death. But oceans are critical for climate and just sustainability in general. So I think that maybe there should be some sense of urgency with respect to also ocean discovery.
There's absolutely a huge sense of urgency, and if you don't feel that, you should, because, yeah, absolutely.
Because the oceans look so pretty, and there isn't, they sort of have this sense of, well, it's beautiful. What could be lying underneath? And we know, also from the Tara visits and other oceanographers that have come through Radcliffe, there's a lot that to say that there's a lot we don't know is completely underestimating what we really don't know.
That's true.
So Joel, I had a question for you with respect to the big data aspects of all the discoveries, because I myself dabble a little bit in big data from a cancer perspective. And even when we look at data that was generated by TCGA, which is the Tumor Genome Atlas, where in my own disease that I studied, it was treated as this composite disease, and oh my goodness, people were shocked that when we looked at the molecular, we looked under the hood, so to speak, the molecular subtyping said, oh my goodness, this is at least 4 different diseases. Now, I wasn't shocked by that, because at the time, you relate to, you go with what you have, which is histology. So increasingly, we're finding out more and more with respect to every type of disease, the 4 types of diabetes, there's at least 4 or 5 types of endometrial cancer, there are 5 types of ovarian, and I'm sure as you start binning these diseases, not just by their genetics and their genomic profiles, and you start adding in lifestyle and environment, which we're going to get increasingly, not just from the medical records, but we already have in hand from many of these studies. My point is, we're going to be so ahead of these brilliant physicians whose job is really, to be fair to the physicians, is to blunt the bleeding. It's really to just take care of the problem at hand. So I'm not surprised that your dermatologists and your GI doctors were sort of working in silos, because they're so trained to, that's their job. You have a GI problem, you work on the GI. You don't think about the inflammatory immune response. So it's not just a statement. The question is, we have all, because this also plagues what I do, we have all this data, and you showed some brilliant networking. How do we translate that to the clinic, especially in light of the fact that Mount Sinai also has this 360 kind of lab? I mean, what do you tell the patient or this person who comes in and says, well, we found X, Y, and Z, but there's really not much we can do because therapeutics or the treatment hasn't gotten there yet? I'm sorry that was so long.
Yeah, it's big. Yeah, I'll try to be brief. It's a big question. I think one is to try to, the health system itself is not set up, to sort of support this type of thing. So what we're trying to do with Lab 100 is to rebuild the health system, so it can support this type of thing. And one, to bring the science of precision medicine to precision health and wellness, right? Because pre-disease, it's a lot easier to prevent a lot of these diseases. But it's really about, I think the physicians will be open as long as you equip them with the tools, right, to act on the information and act on the information at the right time, right? Because they're busy. I'll give you an example. We've known that there are certain genetic markers pharmacogenomics that should influence prescribing of medications, right? So,
Right.
And we actually have 50,000 of our patients that are sequenced, and the existing electronic health record software can't even connect a genetic marker of the patient to the time when the drug is prescribed and alert the physician, that we actually had to spend $4 million at Mount Sinai to build our own software to do that, right? So I mean, there's a last mile problem of connecting what's
Right.
known to the clinician, but in a way that it's at the right time in context of giving patient care, and that's a big challenge.
Right, and I think that's the infrastructure. It's sort of like we need to build this infrastructure.
And I'll just add very briefly, but there's going to be some things we're never going to get the required evidence on, so I think we also need to empower patients more to collect evidence on their own. So I'll give you an example. Is with my Crohn's disease, I asked my physician, what should I or shouldn't I eat? And he'll tell you, well, you eat whatever you want. And you think, that's insane. Well, there's never been a randomized controlled trial on all these foods to tell me what you should and shouldn't eat in Crohn's. And so you're saying, like, I can drink beer and donuts or have a salad, and from the perspective of medicine, it's the same thing for my disease. It's insane. So one of the things we're doing is building a N of 1 clinical trial software so patients can start running rigorous clinical trials on themselves and collect evidence on these lifestyle things. Yeah. So, yeah.
So I'm going to be a little bit of devil's advocate
Yeah.
on that. So that would assume that the patient actually knows the implications of their findings.
Yep. Oh, yeah. So the hardest part about building that software was not the statistical engine that calculates it. It was the user experience.
Right.
Getting someone to understand a p-value and an effect size. Well, we probably spent, a year on that part of it alone.
Easily.
Easily. So that was super hard. And I'm not claiming that we figured it out, but that's super hard.
Yeah, I think that's a general concern that I have, is that when we put out all these, we construct these algorithms and predictive scores, we are assuming that the general population understands what risk is.
Yeah.
And that's not to undermine the intelligence of the general population, but I have colleagues that don't understand what a p-value of 0.05, and they're brilliant. So I guess it's sort of a process, right? We're in a situation where we have to educate low, high, and the gradient, as well as I like to say, we need not only maybe we don't need the depth, but we certainly need the breadth to understand a lot of this big data output.
Absolutely.
Okay, so I think at this point we should open it up to the audience, because I can stay here all day and talk to these wonderful people. There's a microphone that's coming. I would ask that when you ask your question, please pose it in a question. And please state your name.
Big group.
Nice.
Hi, I'm Jo Solid, and I'm a sleep researcher. So this question is for Joel. We know both diabetes and Alzheimer's have relationships to disordered or insufficient sleep. I wondered if your islands included sleep anywhere in this evaluation. We know the brain has no lymph system, but during sleep, the toxins do drain. We don't sleep, the toxins don't drain.
Yeah.
So what do you think?
So we don't currently have that data on patients. We're actually conducting a study using wearable sensors and things like that to measure sleep and incorporate this. And yes, circadian effects we know are huge, and not only influencing the metabolic system, the immune system, and we simply don't have that data yet on a large-scale population, but we need it badly.
Yeah, big piece.
Yeah,
Thank you.
thanks.
Hi, Sally Sieber. Another sort of general question, and that is, those of us who got the early polio vaccine before about, 1973, also got SV40. And I heard a really fantastic seminar, probably in the late 70s, from Germany, where they track all cancers. And they could start to see if there was any difference between people before who got the early polio vaccine versus the other one on various cancers. And one of the things they were hinting at is there might have been some stuff on some brain type cancers. Have you heard any updates or looked at your data with those of us who've gotten SV40?
I do believe SV40 showed up I can't recall off the top of my head, but of course the evidence of the relationship between viruses and cancers is growing day by day. I'll note that one of the genomic features we found that related to the viral activity in Alzheimer's are these things called G4 quadruplex domains. But the only important thing to know is that those are actually being targeted by cancer therapies right now because there's virally driven cancers that target those, and we're actually looking at using those in Alzheimer's as well. So,
Okay,
yeah.
thank you.
Thanks.
Christine Borgman, we study data sharing and reuse in the sciences, and we have found in oceanography, biomedicine, astronomy, and elsewhere that one person's signal is somebody else's noise, and they often don't agree on what are the data in the first place. So many of the effects that you're talking about disappeared at those early stages when they threw away the outliers and did the data compression. How do we think about this better as far as how much are methodological issues we should be training for, how much are statistical, how much are just the equipment, like you said, that you pick the data up, or even the software that threw away those signals because they thought they were noise?
That's a huge question. So I think for sure there's some standardization of oceanographic methods, and I'm sure this applies to other fields too, that we could get on board with, and people are beginning to do that, and that is wonderful. So I totally support those efforts, and then I think we need to get better at having people embrace that noise and not get rid of outliers, like you said. We're super trained to be risk adverse, and yet we know just from sitting here this morning, right, that that's a bad idea. Those outliers have a lot of power to them, and we also need to start embracing negative results, right? Because if we're not publishing those, then we just repeat those same experiments over and over again. So it's a big issue I think we need to deal with.
I'll just add very briefly, one of the things that the Accelerating Medicines Partnership for Alzheimer's did is the program officer required that the rawest form of all data was put into the public data sharing repository. Because the genomic data, the imaging data, right, they go through several steps of transformation. So that was very prescient, and move on her part to and not being a data scientist, she knew it would be super important to get the rawest form possible because of this. Yeah, yeah.
Thank you.
Hi, Ned Bacon. Joel, I wanted to ask you, have you any thoughts about the increased, maybe there isn't an increase, the susceptibility to these viral effects with aging?
Yeah, definitely. I think the ultimate story of the viruses and Alzheimer's are two things. One is that innate immune activation is bad in the brain and neuroinflammation, which we've kind of known. And in fact, the genetic association studies for Alzheimer's have, over the past couple of years, have pulled up a lot of immune markers focusing on this immune cell in the brain called the microglia, where the neurologists are kind of like, oh, why are all these immune signals coming up in the GWAS? This is weird. What do we do with this? But the other side of that is then immune senescence, which could be explaining why, aging of the immune system. Why are these viruses becoming active, in the brain at a certain stage of life? Is it, so, and then there's potentially a role for immune senescence in the activation of these viruses as well, but, still pretty early days. Yeah.
Hi, great talks. Janet Rich Edwards, I'm an epidemiologist, and one of the things my team has been studying is trying to sort of bust open the silos of the way we think about bodily systems. So we correlate the reproductive system with the cardiovascular system and find some new connections there. You mentioned the way that NIH is organized by bodily system. I suspect we're going to look back on this and say, this is such an archaic, quaint way of thinking about the body, that there really aren't such systems. I was intrigued that you mentioned at least one instance of them encouraging you to go across the omics, and wonder if you have any comments about how that sort of silo-busting perspective can be encouraged, perhaps revolutionarily, across all of NIH. And I don't know if NSF has similar issues.
Yeah, NIH is a big, complex place, obviously. I think, again, the folks in the National Institutes of Aging are very forward-thinking. In fact, what they often do is they put me on study sections and they say, go cause trouble. As actually the program officer quietly says, go cause trouble. And the evidence is just mounting more and more. So we actually have a paper under review right now where we do this sort of network analysis across all tissues in the body and looking at diabetes and autoimmune disease and Alzheimer's and all these diseases that have comorbidities. And you can actually find this one master sort of connector in monocytes that actually connects all these different things. So I think it's just a matter of time.
Wonderful talks. My name is Robin Barnes. I'm a retired primary care physician, and we don't just staunch the bleeding. Actually,
I knew I was going to offend many people, but I'm on limited
but
time
in
here.
fact, that brings me to sort of a big speculative question that may or may not be answerable. So part of what's important about medicine, or about particularly primary care, which is longitudinal, and you have chronic relationships with patients whose diseases come and go and so forth. How does what you're doing interact with that? And the bigger question is, how is this going to change the healthcare system as a whole, which, as you know, is not just medical? Just a couple of thoughts.
Ooh, that's a big question.
Well, that's what.
Yeah,
That's
yeah.
basically.
What's that?
That was basically the big question that I asked.
Yeah.
It's just huge.
Yeah, it's huge. So I think the way to do it is, unfortunately, is to sort of restructure the health system fundamentally, and that's actually, so I'm at Mount Sinai, but I don't spend too much time on this, but that clinic you saw, by the way we designed it, it can be built in a week and a half, actually. And actually we have a smaller version of it that's a kiosk. So our mission is to create a parallel, totally decentralized health system that can be deployed closer to where people live, that then puts major pressure on the existing health system to change. So that's my goal. I tell Mount Sinai my job is to shut their primary care down with my new model. So, yeah.
Colleen Kavanaugh, I follow these questions in terms of how revolutionary your approach has been, that it could be applied to the earth more broadly and from nations to the whole earth. And I wanted to ask how you think you can do this and do it fast. And I have one answer from our Microbial Sciences Initiative symposium with Jim Collins. But
I think this is your question.
Oh.
No, it's both.
It's how to heal the ocean. How do we heal the ocean?
Yeah, this isn't an NSF proposal, NIH.
Well, I'm actually quite excited to see what you're doing and wonder if we can do a similar thing for the oceans, because we often talk about ecosystem health and we have no definition for that. And we just go look for problems, right? And that's what we get excited about. But trying to understand that difference between health and disease, basically, of an ocean basin or an estuary, I think we have data to start to do that, but we need these computational tools and to apply those.
Yeah, I mean, my brain goes immediately to microbiome research, which maps very well the gut microbiome, because it is an ecosystem. And there's this dream that people have that if I can just find the healthy microbes and then give people the healthy microbe, it'll make them healthy. But it turns out that it's a big ecosystem, right? And it's a big garden. You've got to kind of prune and seed and stuff like that. And I think that's going to be so there's, like what my talk said, there's not going to be a magic bullet. It's going to be an ecosystem approach.
No, but what I'm looking for is the bullet for the funding.
The bullet for the funding.
Do it fast. And I can tell you, Jim Collins, who invents, figures out some medical problem, solves it in the lab, and in 2 months, it's translated. His answer was, he starts a lot of companies, makes a lot of money, and that money does it.
Yeah.
There is another big funding groups, like the name Gates, Allen. And this is just, this has to happen, and quickly. For me, in particular.
So what we are seeing is of the people who make a lot of money in tech, they are deploying resources because they understand what we're trying to do, and they are funding.
But who wouldn't understand it?
You'd be surprised.
I'm not going to name names.
Don't name any names. Smart. Yes.
Dr. Fulweiler, sorry, my name is Joshua Roth. I'm a textbook editor, and I have celiac disease, so I'm very interested in the microbiome doctor. Dr. Fulweiler spoke to us about finding passion and getting and building teams. And when I'm an amateur astronomer, so I think about amateur scientists, amateur number crunchers, and people who do it for the love, who may not have had it happen in their life to be able to do it for a living or to have a more conventional institutional involvement in science. So the question I wanted to ask each of our speakers in the present session is, are there opportunities for citizen science, for amateur participation, for joining a community of people who do anything from spit in a vial or take a cup of seawater for you to actually help you with the sense making?
So there are absolutely groups for citizen science, and I think you've hit on something that's really fundamentally important, and that is that scientists only see sometimes the things that they're going, and they're maybe just going for research queues for a few weeks, or I'm going down there to study one bay and I'm there infrequently. There are groups that are out in the water, like fishermen, all the time, and there are groups now that are working specifically with people that are on the water, for example, or are surfing or kayaking or whatever it is. There's lots of groups to join for that, and I'm happy to give you examples offline if you would like, but there's watershed groups, and for sure, I think that citizen science can provide a whole new set of eyes and knowledge that we're missing, as well as I think you're probably going to allude to in a second, this idea of being able to capture data and provide it to scientists to sort of do the analysis on.
Yeah, so the trend we have here, of course, is that powerful measurement technologies become rapidly commoditized and distributed out into the consumer realm. Genomic sequencing's falling precipitously. It's easy for anyone with a credit card basically to start sequencing something or bring your minion onto the boat. And wearable sensors are increasing in fidelity. And you may not be aware, of course, that if you ever heard of Apple Research Kit, Apple has a software built into iOS to conduct distributed clinical trials, which isn't quite citizen science yet because you need an academic institution to partner with. So it's happening. And I want to point out there's someone at Mount Sinai named Jason Bobe who has this resilience project, which is a citizen science project to get people to submit biological data to find out if they're resilient to diseases. For example, could we find someone who has ApoE4 at the age of 80 and is cognitively intact, right? So, yeah.
My name is Ricardo Cortez, a retired chemical engineer. I have two questions. One is on the slide that you show nitrogen removal that changes through time. Was that because the data that was in the early ages of the data collection was wrong, or because of climate crisis has changed the chemistry of the ocean and basically reversed the path? And the second question is, if the idea of the viral species is the causation of Alzheimer's disease, and virus multiplying so fast, why is the Alzheimer's disease so slow to develop?
So those early data and were not incorrect. And I'm happy again to talk about this offline. It's a little bit of a long story, but basically the climate warming that we've seen in coastal systems is changing them in ways that we would not have expected. And that is what's been driving the switch that I showed you.
Yeah, I'd say on the viral side, again, there is no cause of Alzheimer's. There's causes of Alzheimer's that all come together in a really complex way. So I didn't note about these viruses, which is important to note, is that they're retroviruses, so meaning that they can integrate into the host DNA. So what we think is likely happening is that so the exposure could become very early in life, and that these viruses are lying in wait in the genome of the neuron, basically, and they're becoming activated later in life. Why they're becoming activated, we're not totally clear, actually.
I'm Paul Ray. I'm just a science enthusiast. But my question is kind of related to his on the global warming and the coastlines, but mostly about the coast height. So, is there studies, and I'm sure you're busy enough trying to figure out what's going on at this moment, but are there studies on if new sediments, if suddenly there's a new tidal flat, that's new sediment, how that differs than an older sediment that's buried? I mean, the history is hard to tell. The coastline is buried under water. But anyway, that's what my question is.
Yeah, so I think what you're getting at is this idea of trying to understand potentially how nitrogen removal capacity changes in sediments over time. Yeah, and there's some really neat ecological theory on this, but basically what you tend to see is really early sediments. You see nitrogen fixation, so that arrow going down, basically the sediment's making nitrogen, the bacteria in the sediment's making nitrogen. And then as you build up organic matter or food for the bacteria, that will switch and you'll start to remove the nitrogen. And there, of course, are caveats to that, but that's the general trend.
I think that was our last question, and there were many questions, a testament to how great both lectures were. And I'd like everyone to please applaud both Wally and Joel.
Well, I would like to thank Mac and Wally and Joel for just carrying us to what is almost lunch in such a beautiful way. And so let me just say one more thing before we let you go. So actually, a few more things. The first one that I forgot to say before is there is a Twitter stream for this. It's hashtag the Undiscovered, and there's a lot going on there, so you can recap what you already heard, and you can go contribute there also. Now, there is lunch, and I mentioned earlier that there's a poster session, and this poster session is dedicated to ideas about how we can incorporate the kind of thinking that you see today in the talks into education, K-12 education and education at other levels. And I just wanted to show you because Stuart showed kind of a draft version of something just like this. One of the posters, which I admit to having something to do with, is about how Isaac Newton got the information and the ideas that he needed going all the way back to ancient Mesopotamia. And if I just let this scroll across the screen, you'll see that that is not the neat arc, okay? And that is the cleaned up version. And if I just show you that again, I want to point out at the left, those 3 circles you can't read. One says a step toward Newton, if it's green. The one at the bottom says wrong or unhelpful ideas, failures, I suppose we should call them. And the one in the middle is the most important, that says partial credit. Okay, so Stuart pointed out early on that nothing is ever totally right, and all of the brilliant work that you heard here is not totally right. It's mostly right, and it's helpful, okay? And so the idea is how do we communicate this complexity without giving people the idea that scientists don't know what they're doing, okay? So that's the point. Thank you very much.